About 20 years ago, Francis Crick, the co-discoverer with James Watson of the structure of DNA, wrote a book called The Astonishing Hypothesis. The astonishing hypothesis is the idea that every one of our thoughts, beliefs, memories, and experiences consists of neurophysiological activity in the tissues of the brain. That is, there is a material basis for intelligence, for emotion, for consciousness. The brain has been called the engine of reason and the seat of the soul in recognition of the fact that, as best we can tell, every twinge that you feel, every bit of deja vu, every plan of what you're going to do tomorrow, every mental image, the experience of fear, of depression, of falling in love, every one of these is caused by or consists of activity in the tissues of this remarkable organ in the skull. This is a picture of my brain. It is a magnetic resonance imaging depiction of my brain. I was a volunteer in an experiment, and I got to take this home as a souvenir. And I've got to say that the first time that I saw it and recognized all of the structures that I had read about and taught about from textbooks all these years, I did get a little bit of a chill. I don't know what I expected, what the alternative would have been of seeing all of the standard parts there in black and white. But still, to think that everything that makes me me consists of wiring and chemistry in that unprepossessing organ still fills me with astonishment and wonder, even after having taught it for many decades. The brain is widely recognized as the most exciting frontier of science today, and the relation of mind and brain continues to pose the most profound challenges in modern intellectual life in philosophy, in politics, in the arts, and in our understanding of the human condition. I'm going to try my best to give you an overview in the next hour and 15 minutes or so of this amazing organ. You're not going to walk out of here experts in neuroscience by any means, but I hope that I will have introduced enough concepts that if you read about the brain in the science section of the newspaper, or you see it in a science documentary, you read on, as I hope many of you will do in textbooks or popular books, you'll have an orientation as to what the basic principles are that allow the brain to work. The brain, as an organ of the body, is made out of cells, just like every other organ. In the case of the brain, the central computational unit of the brain is the neuron. A neuron is a brain cell. Like all brain cells, a neuron has a cell body, including a nucleus. But what makes neurons unusual is that they are shaped in a way that allows them to carry information. They have a number of input processes called dendrites. This kind of looks like a tree, and dendrite comes from the Greek word for tree. They've got a long output fiber called an axon. The axon ends in a number of axon terminals which make contact with other neurons. The axon in many neurons is coated with a number of fatty cells, giving rise to what's called a myelin sheath. The color of the myelin sheath is a kind of glossy white, which, as we will see later, gives rise to the term white matter as contrasting with gray matter that we'll soon come up with. This is a diagram from a textbook. Now we have an amazing ability to visualize elements of the brain and their interconnections. This is an actual neuron grown in culture. Since none of us is a windowless monad, a brain in a vat, we don't just think thoughts, but our thoughts are connected to the world, the brain has to have some way of receiving information from the world and then in turn acting on it. And the three basic ways of thinking about what neurons do is to think about the input, the output, and the computation in between. The input, the structures that convert energy coming from the environment into neural activity, are called receptors. And at the output we have neuromuscular junctions. the computation in the brain is done in synapses. A synapse is a connection between one neuron and another. And I'm going to explain each of these three right now. This diagram shows how your brain allows you to have commerce with the world. A receptor is a special kind of cell that can convert some kind of energy into neural impulses. That's what gets the entire process of consciousness started. For example, your skin is studded with a variety of receptors that respond to temperature or pressure and that convert that energy into neural impulses that proceed along the dendrites into the cell body and then out through the axon. Here you've got your spinal cord, which is, together with the brain, forms the central nervous system. Here we've got a synapse between a sensory neuron that goes from the skin into the spinal cord. It makes the axons of the sensory neuron form synapses with the dendrites of an interneuron in the spinal cord. The axon of an interneuron in turn makes connections with the dendrite of a motor neuron. The motor neuron then proceeds out through the spinal cord, down your arm, and makes contact with your muscles with another structure called a neuromuscular junction. This completes the connection between the brain and the world, because at a neuromuscular junction, when neural activity reaches the end of the axon, it causes your muscle fiber to contract. The central nervous system consists of the brain and the spinal cord, in distinction to the peripheral nervous system, which is the network of nerves that innervate your body and allow you to feel and to move. This is going back to the picture of my brain. Here we've been talking about the spinal cord, which connects you to the world. Here is my spinal cord going up through my neck. Now, you'll see that it enters the bottom of the brain. This obviously is the skull. And there is a set of structures here that form the core of the brain. The messages from the body go in through it. The messages down through the muscles go out through it. And this structure, because it's kind of like the stem of a fruit, is called the brain stem. You'll see that the spinal cord has a kind of thickening as it enters the base of the skull. This thickening is called the medulla. There's also a band of fibers that goes across it called the pons or bridge. You don't have to obviously memorize these labels as I'm explaining them, but they are just orient you to what the major parts of the brain are. And at the top, there are two sort of apricot-sized organs called the thalamus. The thalamus is basically the relay station where information that comes in from the senses passes through the thalamus when it goes to the rest of the brain. Information from the brain that then commands the muscles to move typically goes out through the thalamus, then again back out through the spinal cord and out to the muscles. So all that I've been rushing through right now is just to orient you to the position of the brain in the rest of the body and how it communicates with the world. For those of you who have a further interest in the structure of the brain, particularly if any of the information in today's lecture whets your appetite, but you feel a little bit confused as to the geography of the brain being a very complicated three-dimensional structure, one that even people who have dealt with it all their lives sometimes get lost trying to figure out how the different brain areas connect with each other. I encourage you to consult an app called 3D Brain. You can download it for free as a iOS app, or you can see it on a website. Just Google 3D Brain and it will come up. I've done that for you, because the nice thing about 3D Brain is that it allows you to see the three-dimensional structure in either by rotating it or by peeling off various layers and seeing what lies embedded underneath. Each one of the structures labeled in 3D Brain also has a little bit of a description as to what it does that can serve as a backup for the very quick tour that I'm going to give you this evening. A major set of structures of the brain is lumped together as the limbic system, and that's something you can think of as the seat of motion and motivation. Sometimes scientists abbreviate them as the 4 Fs, that is feeding, fighting, fear, and sexual behavior. I'll very quickly go over them, although the focus of this lecture is on the parts of the brain that allow us to rise above the 4 Fs, namely the cerebrum, the big bulbous gray structures that allow us to engage in rational deliberation. But just a quick introduction to the limbic system. We've got the hippocampus. It literally means seahorse because it reminded a creative neuroanatomist of a seahorse, the man who first discovered it, which is involved in consolidating memories and in investing them with emotion. You've got the amygdala on each side, which is Latin for almond. Again, the anatomists were desperately in search of terminology, so they named structures after whatever they were first reminded of. You've got the hypothalamus, which is a tiny yet vital collection of little nuclei, little lumps, that are responsible for many of the motives and emotions that just keep your body going: regulation of temperature, the control of heart rate, sexual desire, the hunger, thirst. And the hypothalamus is connected to a gland called the pituitary, which is just behind the soft palate in the back of your mouth. The pituitary is bathed with blood and can drip hormones into the bloodstream, which is another major way in which your brain can communicate with your body. The brain, via the pituitary, can secrete hormones that then go to your adrenal glands, which in turn pump out adrenaline, which gets your heart racing, your breathing shallow, your fingers clammy, and so on. And it's a kind of quick and systemic way for the brain to affect the entire body in a hurry. But what I'm really going to concentrate on is the cerebrum. The cerebrum consists of the 2 large conspicuous hemispheres that kind of dominate your view of the brain from the outside. Now, I mentioned that there is a fundamental distinction in neural tissue between gray matter, that is, neurons that collectively are unmyelinated, they lack that white, glistening, fatty sheath, and white matter. But it's the gray matter, often used as a synonym for intelligence, thought, rationality, and here's the reason why. The gray matter corresponds to the cortex of the cerebrum. Cerebrum refers to the entire hemisphere. The cerebral cortex is kind of like the skin of the cerebrum. It's the outermost layer. It's about 2 to 4 millimeters thick. It itself has a very precise structure consisting of 6 layers. And the basic computational unit of the brain is called the column. It is basically a tiny little core of part of your cerebral cortex. Let me give you a picture of one. First, the exposed cerebral cortex. This is a patient undergoing brain surgery who's had a piece of the skull removed, and so you're actually seeing a bit of the exposed cortex right here. Here is a cortical column actually visualized through a microscope. You're seeing about 10% of the neurons here, the ones that pick up a stain, and you can see the beautifully arranged layers. A cortical column is about 1 cubic millimeter, so it's a tiny little eensy-weensy dab of brain tissue, but it contains on the order of 10,000 neurons. This is a computer reconstruction of what a cortical column looks like, and it just gives you a visual impression of how much complexity there is in that tiny little pinpoint of brain. Multiply that by the 3 pounds of tissue that you carry around in your skull, and you get a taste of the almost unfathomable physical complexity of the human brain. By the way that neuroscientists first discovered that the basic unit of the brain was a cortical column, that is something that a bit of tissue that's oriented at right angles to the surface of the skull, is a simple study done in rats in the 1930s. If you expose the brain of a rat, you take a scalpel and you make a bunch of little crisscross cuts on the surface of the brain so that you are, along the surface, the behavior of the rat is barely affected. But if instead you make a cut that is parallel to the surface, then you make the brain, the rat blind or paralyzed or amnesic. So this shows you that the circuitry in the brain doesn't actually run along the surface, even though the cortex consists of the surface, but rather the functional circuitry of the brain is at right angles to the surface of the skull. The fibers go down from the skull into what's called the white matter and then come up again and make contact in another portion of the surface of the brain. I'll return to that soon. Oh yeah, so white matter. The gray matter is the cortex. Looks gray, as you'll soon see. The white matter is kind of the core. It fills the inside of the lobes of the brain. And these are bundles of axons that connect one part of the cortex to another in U-shaped tracts. Now, here's a somewhat unusual way of getting you to appreciate the difference between gray matter and white matter. It so happens that when I was lying down in the scanner, my head was not perfectly straight. I was kind of cockeyed a little bit. And you can see that in the fact that in the cross-section of the brain, this is the section that is right in the middle of my two cerebral hemispheres. There's a space in between them. But in the back half here, you're seeing gray matter. Here, these are, you can see the various lumps and folds. The lumps are called gyri, the folds are called sulci, gyrus and sulcus. But over here, because I was sitting at an angle, it was like the blade of a guillotine actually sliced through my brain over here. And so in the front half of the brain, you're looking at a cross-section, not the exposed gray matter, as you see here, but rather it's as if the beam actually cut through the brain tissue. And you can see the outer surface here of each one of these lobes is gray, and the inner core is white. So that's gray matter in the cortex, and this is white matter consisting of fibers that link one part of the cortex with another. Here is the next slice in the, when you do an fMRI, you're kind of like in a salami slicer that takes a succession of sections through the brain. So over here, the next one down, here the front part of my brain is, the slice went cleanly between my two cerebral hemispheres in the front. In the back, I got nicked, I got sliced like a bit of salami, and here you can see the white matter on the inside and the gray matter on the outside. I'm going to show you now the same thing, but at this point it really is like a guillotine in that now the slices are going to come from the top down. So this is from the kind of the first slice looking down from the top of my scalp, and this is all gray matter. Here's the next slice. You can see the white matter over here and the various gyri, that is the various outward bulges. These are sulci, grooves. Here's the next slice down, showing that the gyri are in turn collected into major lobes. The brain is kind of like a broccoli, in that it's got a bunch of stems which then branch and then branch and then branch. That is, the brain is not just like a lump of spam, or it's not like your liver, it's not a homogeneous structure, but rather it has an internal branching structure of white matter leading to gray matter. And again, here is a close-up showing a few of my gyri with the white matter here, the gray matter on the outside. Nowadays, there are increasingly sophisticated non-invasive techniques of illuminating brain anatomy using magnetic resonance imaging, MRI, and other techniques. Here we have, it's a little misleading because it's obviously not white, but this is false color. It's a color generated by the computer of the white matter tracts in the brain. And again, it gives you a feel for the fact that the white matter is not just meatloaf, but rather a set of fibers that connect one part of the cortex with another. It's as if we dipped the brain in acid, got rid of the gray matter, and all that's left are the white matter connections. This band of fibers over here is called the corpus callosum. It allows the two cerebral hemispheres to talk to each other. Now, the real thinking and feeling and remembering and actual intelligence in the brain takes place at the level of neural networks, that is, circuits consisting of many tens of thousands of neurons connected to each other via synapses. Here again we have a stained slice of the real brain, just showing you some of the complexity that's involved in the neural circuits. In order to make some sense of it, theoretical neuroscientists try to figure out what the brain is doing by modeling the neurons in much simpler models called neural networks, where each one of these circles with an arrow is like a toy neuron, just so you can try to figure out what can be computed in neural networks to understand how the brain is doing what it's doing. Those of you who are going to attend my lecture tomorrow, I'm going to drill down to the level of neural networks and show how the flow of information in these complicated webs can actually do intelligent things. For tonight, however, that's going to have to be a bit of magic, and you're going to have to take my word for it that chunks of brain tissue compute interesting things. One of the key issues in understanding the brain is the contrast or continuum between localization and mass action. That is, our particular psychological functions of the kind that we're familiar with just in living our lives and in dealing with other people, can you localize them in a patch of brain tissue? Kind of like, I don't know if here in Britain they have what we have in the meat counter of the supermarket back in the United States. Sometimes you have a cow with dotted lines that indicate where the flank steak is, and the sirloin and the chuck and shoulder steak and so on. The question is, can the brain also be divided like the supermarket cow into regions that correspond to psychological functions? Now, the most extreme theory, localizationist theory, belongs to the old pseudoscience called phrenology, P-H-R-E-N-O-L-O-G-Y, a popular pastime in the late 18th and middle 19th century. You may be familiar with diagrams like this. Here's a slightly more legible one. According to the theory of phrenology, every characterological trait had a patch of brain that implemented it, such as acquisitiveness, constructiveness, love of the magnificent, spirituality, union for life, love of friends. Moreover, the more a person had this trait, the bigger that part of the brain was, forcing the skull to bulge out at that, over that part of the brain, and hence allowing a phrenologist to kind of reverse engineer your personality by feeling the bumps on your brain and assuming that if you have a big bump over here, that must mean you've got a lot of love of beauty. Now this turns out to be total nonsense. It is now known today mostly in a number of satires, such as Inside Patty's Brain from the New York Times, where she whimsically locates the part of the brain that stores her eBay password from Amazon.com. Number 2 is Directory of Women Thinner Than I Am. Area number 6, John, Jim, no, maybe a B name, Bob, Brad, no, so phrenology lies in the dustbin of obsolete scientific theories. The opposite extreme in this continuum is the theory of mass action associated with the great neuroscientist Carl Lashley, who worked in the 30s and 40s and 50s, the same man who did the experiment of cross-cutting versus undermining bits of brain tissue to determine that brain circuits are U-shaped. Lashley did another famous experiment that's kind of passed into popular consciousness called In Search of the Engram, the engram being a now obsolete term for the neural trace of memory. He trained rats to run a maze in search of food, then cut out various parts of the brain, searching for the part of the brain in which the rat's memory of the maze was stored. And the famous finding was he never found it. That is, there was no single part of the brain which, when extirpated, left the rat amnesic for the structure of the maze. Rather, the more brain tissue he took out, the stupider the rat became. Well, this led to the unphrenology theory, the opposite of phrenology, which is basically that all of the brain does everything. Now, that turns out to be too extreme as well. This, by the way, is probably also the source of the widely repeated but utterly false legend that we all use only 10% of our brain. That probably originated in this experiment. It's completely wrong. But also what's wrong is that something as diffuse as knowledge of a maze, which requires both a mental map of the geometry of the maze and a set of instructions, turn left here, turn right there, is not the kind of thing you'd ever expect to find in one part of the brain. And it turns out that Lashley was far too pessimistic about the extent to which different portions of the brain corresponded to different aspects of what we do. So strict localization is not correct, not just the phrenological version, which is obviously preposterous, but even today in modern neuroscience, all neuroscientists reject the idea that any particular trait, any particular function, such as language, such as vision, such as memory, resides only in one little patch of the brain. There is no single brain area for X. Brain function involves networks where different parts of the brain communicate with each other in order to allow us to do pretty much anything. Also, it's hard to find patches of the brain that do one and only one thing. Finally, as I mentioned earlier, the actual intelligence that your brain accomplishes, the fact that I can ask you a question and I can get a sensible answer back, doesn't depend on lumps of tissue. It depends on that unfathomably complex computation done in the microcircuitry, the microscopic individual synapses and networks of neurons. On the other hand, Lashley's other extreme of mass action is not complete, is also not correct. As you'll see in the rest of this lecture, we can give approximate characterizations of what different parts of the brain do, as long as we remember that they don't act in isolation, but act always in concert with other parts of the brain. As one neuroscientist put it, the brain is globally local and locally global. What does that mean? Globally local means if you kind of look at the brain from a bird's eye view, hold the brain up in front of you, it's not all interchangeable. You can say that this part of the brain does more vision than that part, this one's a little more involved in hearing, that part a little more in emotion, and so on. On the other hand, locally global means that if you drill down to one little part of the brain, you tend to find that it is involved in not just one function, but in many functions. So that's just a general warning. With that in mind, I'm going to now give you a guided tour of your cerebrum, giving you the approximate division of labor of psychological functions among anatomical structures. The first thing, in order to make sense of the brain, is you've got to at least divide it into its major lobes. You saw from the diagrams, the broccoli-like diagrams that I showed you earlier, that the brain is not a mass, but rather has fibers that connect big chunks of cerebrum. And the four main ones are named after the bones of the skull under which they sit. You've got the easiest one to remember is the frontal lobe, because it's under the frontal bone, which is not surprisingly in the front of the skull. You've got the temporal lobe. That's this kind of hand-shaped lobe that sticks out kind of almost like laurel leaves horizontally on both sides of the brain. So-called because it sits under the temporal bone. In turn, so-called because the temporal bone is where your temples are. That's an easy way to remember the temporal lobe. You've got your parietal lobes. Those sit under the, here, under the walls of the skull. Do any of you know what the word parietal means? Have any of you heard of parietal rules? Okay. I noticed that the only hands that have gone up are from people who possess gray hair. And that's because parietal rules are the now obsolete rules on college campuses that forbade men to go into women's residences and vice versa. Parietal rule referring to the hypothetical wall between the sexes, but the Latin word simply means wall, and the same word goes to the parietal bones and the parietal lobes. Finally, you've got the occipital lobe, which is that kind of bun-shaped structure at the back of the head, that bulge. Occipital means ob caput, against the head, and as we'll see, it's a major seat of visual processing. Okay, so here is just another reminder that if all of this is whizzing by way too fast, you can get a clear reminder of the structures of the brain, their three-dimensional shapes, and their functions by going to 3D brain. Now, I've just given you 4 big lumps. I've given you 4 names. How do you make sense of what they do? Is there a way of making sense of it other than memorizing a list of associations between this lobe and that function? Well, there is. And there's a way to kind of make sense of why the different parts of the brain do what they do. The starting point is to pay attention to this major rift here, this major sulcus or groove called the, not surprisingly, the central sulcus, which divides the massive frontal lobe from the equally massive parietal lobe. That is your Greenwich Mean Time, your prime meridian. And it was the first part of the brain whose function we really understood. Pay attention in particular to the strip of tissue lining the front or anterior part of the central sulcus and the strip of tissue behind it. The discovery of what these do was made by a Canadian neurosurgeon named Wilder Penfield. Any of you who visit Montreal will see that there's a major boulevard named after him, exploiting the remarkable fact that the brain itself does not have any pain receptors. It has no sensitivity to pain. If I were to remove your skull, as long as I anesthetize the skin, I could touch your brain and you wouldn't feel a thing. Which means that people who undergo brain surgery are often fully conscious, other than the anesthetization of the skin. And here, so you can stimulate parts of the brain with an electrode and ask the patient, what are you feeling? What is the state of your consciousness? And here is a very old film clip of some of the first of those studies done by Wilder Penfield.
Observing the living brain during surgery was pioneered by Wilder Penfield, a Canadian neurosurgeon who first tried it in the 1930s to treat epilepsy. Once the skull is opened, it is possible to touch the brain without the patient feeling any pain and to talk to the patient about his sensations. Penfield found that his electrical probing could produce, for example, a ticklish nose. Working with patients in this way, he plotted the first sensory maps.
Okay, so a sensory map consists of a depiction of what part of what's called the somatosensory cortex or somatosensory strip underlies the sense of touch in various parts of the body. So here we've got the somatosensory strip. It's the foremost gyrus in the parietal lobe, just behind the central sulcus. And the artist here has drawn the different body parts that correspond to various stretches of cortex. That is, if you stimulate that bit of cortex with an electrode, what part of the patient's body tingles by his own verbal report. You can see a couple of interesting things. First of all, every part of the body has a representation in the brain. By the way, when I was an undergraduate student, mysteriously, the somatosensory cortex had no representation of the genitalia. So I always wondered how it was possible to have any sensation in the genitalia if there was no brain region that registered them. Turned out it was just prudery on the part of the textbook manufacturers. There really is a part of the brain in which the genitals are registered. Now notice also that the different parts of the body are represented in different sizes, which corresponds to how much brain is devoted to that amount of skin, or the skin in that part of the body. Now, this dismembered body here is sometimes called the sensory homunculus, the little man, and it's the greatly exaggerated lips and tongue and a hand, especially the thumb, compared to the minuscule amount of brain tissue given over to the trunk, the hip, and the knee, shows that the parts of the skin in which we make the finest distinctions, such as the tongue and the lips, which we need for articulating speech sounds, the fingers for manipulation, need lots of brain in order to register those fine distinctions. In contrast, parts of the body where you can't do a whole lot of precision movement with, such as your back, are represented by a tiny little bit of brain. And you can even do an experiment that confirms this. If you can't see what part of your back is touched, or even your arm, if you close your eyes and you try to determine whether someone is touching you with 2 fingers or 1 finger, or where exactly on the arm or the back you're being touched, you will have a very hard time doing it. Whereas for your fingertips, of course, the sensitivity is so great that you can read Braille or get a lot of information about the shape and texture of an object. Now, in the strip on the other side of the central sulcus, if the surgeon stimulates that part of the cortex, you get muscle twitches. And depending on what part of the cortex is stimulated, different muscles will twitch. If you then map out the different parts of the body according to what part of the brain stimulates them, you get the motor homunculus. And once again, now here there are no genitalia because there's not a whole lot that you can actually do with your genitals. There's not a whole lot of different muscles and joints in there. On the other hand, for your lips and jaw, which you can manipulate quite precisely in order to speak, you've got a massive swath of brain tissue devoted to them, likewise for our amazingly dexterous prehensile hands. This cartoon here looks a little bit like Mick Jagger. Is another way of depicting the motor homunculus, where the size of the body parts, such as these massively outsized hands and lips and tongue, correspond to how much brain is devoted to those body parts. Here you see the spindly arms and legs and almost non-existent trunk, showing that you don't need a whole lot of brain to go like that, but you do need a whole lot of brain to go like that or to do what I'm doing now. Okay, now why did I start with that part of the brain? It's not just that these were the first areas of the cortex to be precisely mapped, thanks to Penfield. But if you think to yourself where the parts of the brain are that correspond to the 5 senses, then that's a way of making sense of a lot of the more abstract functions that are also computed by the brain. So we've seen the primary motor area, the primary somatosensory area. I mentioned before that vision is first registered in the occipital lobe at the back of the skull. Audition, that is hearing, its primary sensory area, that is the parts of the brain that register sounds, are in an area called the Heschel's gyrus, or the primary auditory area, which is tucked into the top surface of the temporal lobe. If you use these as kind of landmarks, motion, touch, vision, hearing, then you can make sense of all these other areas in between them by thinking that areas of the brain that are near the sensory and motor areas kind of do what is relevant for those sensory areas. Oops. And seem to have lost our... It's back, very good. So for example, I'm going to return to this later, but you've got vision over here, you've got the body represented over here. What do you think all of this brain area, the parietal lobe in between them, is doing? Well, as we'll see, that is responsible for your abstract sense of space, of kind of where things are, which makes sense when you think that the world is out there. It doesn't really care whether you experience it through touch or through vision. Space is space, objects are objects. And so the part of the brain in between seeing and the body is the part of the brain that allows you to keep track of where things are in the world. Likewise, if you think about that learning to read involves an association between a visual representation on the page, namely the letters, and the sound of the word, well, whoops, here we go. Vision starts here, audition is over here, and therefore it comes as less of a surprise that your knowledge of visual word forms, your ability to read an English word, is in some of the brain area that's between the visual area and the auditory area. So that's a way, once you, through just brute force memorization, know where the 5 senses are, the other higher abstract forms of computation kind of fall into place in terms of their proximity to them. Now, this is a depiction of the primary motor areas, that is the motor strip that you saw, the primary sensory areas, that is touch, hearing, and audition, in a number of animals that are related to us via evolutionary relatedness, but that we tend to think of as varying in how much kind of higher order cogitation they do. I mean, rats have not been known to do a whole lot of philosophy. And if you look at the pink and blue areas, which correspond to the parts of the brain that register the senses and that control the muscles, versus the areas depicted in gray, sometimes called association cortex, the parts of the brain that are involved in actual thought and memory. You can see that if you're a rat, most of your brain consists of taking care of the senses and the muscles, whereas for smarter animals like a cat, still smarter animals like a chimpanzee, and at least by bragging rights, the smartest of all, the human, most of our brain is not devoted to the five senses. We've got lots of extra brain tissue for doing philosophy and history and sports and gossip and music and all kinds of other stuff. Which is kind of the neuroanatomical basis of this cartoon. We've got our lobe-finned fish thinking eat, survive, reproduce, eat, survive, reproduce, eat, survive, reproduce. What's it all about? That kind of is reflected in the fact that we've got all of this brain tissue that allows us to think what's it all about and doesn't have to worry about eating, surviving, and reproducing. Now, I mentioned that areas of the brain that subserve abstract functions tend to be near the parts of the brain that underlie the associated senses. And a good example of that is an area in the left hemisphere called Broca's area. It's approximately around here, which, as you will see, is essential for the generation of articulate speech. And not coincidentally, it's not too far from the part of the motor strip that controls the lips and the tongue and the jaw and the vocal cords. Well, what happens if someone suffers a stroke, that is, a blood vessel is obstructed by a clot, a part of the brain is starved of oxygen, that brain tissue dies. Here's an example of a patient who suffered damage because of a stroke, and you'll now see leaving him with a condition sometimes called Broca's aphasia, typically associated with damage to Broca's area.
We have a tape of such a patient demonstrating this agrammatism. He has a Broca's aphasia, now 5 years post-onset, and also carries a diagnosis of apraxia of speech. In this segment, his favorite topic is under discussion, restaurants.
All right, so tell me about the meal you had at Max's.
Oh, yes. Way over the 2 weeks later.
You didn't go there?
No. Last, hungry. Hey, that's what I like. Max. No, but
That's what you were thinking about.
yeah. System.
Are you thinking of Pat?
Yeah, Pat.
Your wife?
Yeah. Why?
So you're going to call her up and say, Pat, let's go to Max's.
No. Here, that's okay. 2 weeks. Little more? Nice.
But as you can see, he is not paralyzed in terms of being able to make sounds, and nor has he forgotten individual words in the English language. But what he has lost is the ability to string them together into grammatical phrases and sentences and to articulate them fluently. Abstract functions that we tie to Broca's area, thanks to patients like him, and Broca's area, again, not coincidentally, is close to, but distinct from the areas that actually control the mouth. Let me turn now to the visual system, which is the part of the cerebrum that we probably understand the best, thanks to the fact that unlike language, which we could only study indirectly via, for example, patients who have suffered strokes, because there are no animal models of language, the visual system of a monkey is very, very similar to the visual system of a human, and at least under the current ethical principles that allow us to experiment on other primates such as monkeys. An enormous amount has been learned about the monkey visual system, and as you will see, much of it applies to humans as well. Now, the information gets into our visual brain through, not surprisingly, the eyeballs. This is a top-down view. Here are the eyes, here are the occipital lobes, and through a clever bit of wiring. I mentioned that the, maybe actually I didn't mention, but anyway, here is a fact. Brain organization is contralateral. That is, your left hand is controlled by your right hemisphere, your right hand is controlled by your left hemisphere, and so on for the rest of the body. You might think that because of that, your right eye would be connected to your left hemisphere and your left eye to the right hemisphere. And indeed, that is the way it's done in some fish who have eyeballs on the side of their head kind of looking outward. But because we're primates and our eyes have migrated to the front of our face, both eyes look out on almost the entire visual world. So the left side of the world and the right side of the world both go to the right eyeball and both go to the left eyeball. But thanks to some clever wiring, which I won't talk about tonight, the information for each eye diverges so that information about the left visual field, that is everything from here leftward, goes to the right hemisphere. Everything from here rightward goes to the left hemisphere, which is a much more sensible arrangement given that the body, which is represented contralaterally, has to deal with space, which doesn't care which eyeball it comes in through. What the brain cares about is where in the world things are. The primary visual area is just where vision begins. It's the first way station, the first arrival point for information when it comes in from the eyes through the thalamus into the cortex. And an interesting feature about the primary visual cortex in the occipital lobe is that it represents the world topographically, which is to say that adjacent parts of the world are represented in adjacent parts of the brain, almost as if you had a distorted picture of the world superimposed as activity on the surface of the brain. This was discovered in an ingenious experiment several decades ago in which a monkey was shown a flashing bullseye like that. The monkey was injected with a radioactive form of sugar. The neurons in the brain that were more active, that were firing more intensely, took up more sugar. The monkey was, as it was looking at this flashing bullseye, was sacrificed. That's a euphemism for killed. And the brain was quickly extracted and placed against a photographic paper so that the radioactive parts of the brain actually lit up the corresponding portion of film. And what you found is, I don't know how easily it is to see because of the dimness of the projection, but these stripes here along the surface and then these perpendicular stripes actually correspond to the last thing that the monkey saw before it died, showing that geometric patterns that you see are actually mapped onto geometric patterns of activity on the surface of the visual brain. Now, decades later, we had the technology to do a kind of similar experiment in humans using much less drastic technique, no radioactivity, no sacrifice. Thanks to the increasing sophistication of fMRI, functional magnetic resonance imaging, which unlike the MRI images that I showed you earlier, which were basically high-tech X-rays, just showing you the anatomy of the brain, the F in fMRI stands for functional, which means that it gives you a map of what parts of the brain are more active as the person owning the brain or being the brain has some kind of experience. In this case, it's a similar experiment to the one with the monkeys, but the person watched an expanding ring, then an expanding ring, then an expanding ring, sort of like dropping a pebble in a pool. And the computer-generated image of the back of the brain, here we have the occipital lobe, was depicted in false color, so that anything you do about that static? When the center of the visual field was stimulated by the ring, the computer painted the brain image magenta. When it was a little bit farther out, it depicted it in orange, then yellow, then green and blue. So cool colors corresponded to a ring that was in the periphery of the visual field, warm colors when the ring was in the center. And as you can see, there's a nice gradient of warm to cool along the surface of the brain, corresponding to the actual thing that the human being saw. So in both monkeys and humans, you have this topographic mapping in the primary visual cortex. This is a diagram of the brain of a monkey, which, since the cerebral cortex is so convoluted, it has those gyri and sulci, those grooves and bulges. It's actually very hard to visualize what's next to what in the brain because of this wrinkly structure. Here you have the equivalent of if you could take a monkey's brain, monkey's cortex, and somehow fold it flat on a table. Now to do that, you'd have to make a lot of cuts, as you would for any three-dimensional surface, flattening it onto a two-dimensional surface. Here's the monkey's brain. Here's the back, the occipital lobe. Here's the front. There's the temporal lobe. This is V1 is the primary visual cortex. It's the area that I've been talking about so far. All the other areas indicated in black and gray are other visual areas in the monkey's brain. And so I'm showing you this just to get you to appreciate that seeing is not just a matter of having a topographic projection in the back of the brain. That's just the first step in a complicated set of computations that allow us to see. This is a schematic diagram of what you just saw here, the visual brain, where each one of these rectangles corresponds to a patch of brain, and the lines correspond to bundles of axons or white matter connecting them. There are some 50 different areas in the brain that allow you to see. Which leads to the question, what are those areas doing? Why isn't it enough to have a kind of video representation of the world playing in the back of the skull? Well, the answer is there isn't any little man watching the pattern of activity in the back of the skull. If that's all we had, it would be pretty useless. We have to ultimately know what's out there, who we're talking to, where they are, what are the surfaces, what are they made of, and in order to get that more intelligent kind of computation out, you need a lot of subsequent processing. They begin with the secondary visual areas, the areas just surrounding the primary visual cortex, which are also essential in vision. An area called V4 seems to be disproportionately involved in the perception of color, for example. So if you imagine a coloring book, but without the black lines, just there's redness here, and there's blueness here, and there's blackness here, area V4 seems to be responsible for that. If you have a patient, a neurological patient who suffered a stroke that destroys area V4, you have a strange condition called a chromatopsia. It's not the same as color blindness, because a chromatopsic patient sometimes can distinguish red from green, but just the subjective experience of the world having its ordinary colors has been lost. They will sometimes report that they have lost their appetite because food no longer looks like food, but looks kind of gray and slimy. They will often be unable to look at other people or to be sexually attracted to other people because everything looks like kind of, one patient described it as dead rat flesh. That is the flush and intrinsic color that allow us to appreciate the surfaces of things as in terms of what they represent is lost if area V4 is no longer functioning. There's another secondary area. There are many of them, but I'm just going to give you two examples to show you how visual computation becomes increasingly abstract as you move up the chain of processing from primary visual areas to sensory ones. If area MT is damaged, then you get a condition sometimes called motion blindness. Now again, the patient is not blind. They can see objects at particular locations, but they can't actually see the fluid motion that connects one location to another. So a patient with motion blindness will see tea flowing out of a teapot as if it was a solid icicle. Or they'll have a lot of difficulty navigating the world because as they're standing on the pavement, they will see a car at the end of the block, and the next thing they experience is the car right in front of them. So they perceive change of location, but they don't perceive motion. Even though one definition of motion is change of location, but even though logically those may be the same thing, psychologically they're not the same thing. And motion as a particular quality of experience requires its own dedicated brain area, area MT. Now from the primary to the secondary areas, then you then get a major bifurcation of information processing in the visual system into what are technically called the ventral and dorsal visual systems. Ventral means belly, dorsal means back. Because our skull has taken a right angle bend in the course of evolution, that is, we're no longer horizontal like dogs and monkeys, but we're vertical. So ventral for us, unlike every other mammal, actually means dorsal means top and ventral means bottom. The terms have sort of been grandfathered over from other mammals. But the terms that I like better for these two major divisions of the visual system are the what system and the where system, because they seem to specialize in recognizing what things are or where things are. Now, this is significant because in ordinary consciousness, we don't appreciate that these are actually neurobiologically separate processes. I see a camera over there, and the camera-ness and the there-ness are kind of fused in my consciousness. I can't just allow the camera to float anywhere in the visual field, and I can't see just anything in that position. They are glommed together one and the same thing. But in the brain, the camera-ness of the camera, or the face-ness of the face, and the fact that there is matter at a particular location are separate processes computed in the what system and the where system. Now, here again is the primary visual cortex in the occipital lobe. Here is the ventral stream going down into the underside of the temporal lobe. Here is the dorsal stream going into the parietal lobe, and eventually making contact with the part of the brain that registers the body. Now, how do we know that? I've just given it to you as a dogmatic statement that the visual information is bifurcated in this way. But once again, it's an example of how you can come to the same conclusion using our primate cousins or with more sophisticated techniques in Homo sapiens itself. So here's a landmark experiment that showed that we have two visual systems, one for where, one for what. And you can see by the fact that this is a sort of a simpler looking brain that we're talking about monkey, not human here. If you lesion this part of the monkey's brain, this is the dorsal stream, the temporal lobe, here's what the monkey can no longer do. The monkey is hungry. It's fed little bits of food that are placed in a well that is covered by a block. The monkey has been trained that in this experiment, that the food is always under the striped triangular block. Okay? It can be on the left, it can be on the right, it's varied at random from trial to trial, but always lift up the triangular block and you'll find a grape. Now, a monkey who has lost that part of the brain can no longer do that task. Even if he's been trained 10,000 times, he will guess at random. The other task, you've got two covers that are visually identical, but you've got a little landmark here, a little cylinder And now the rule is lift up the flap that's next to the cylinder. Well, a monkey who has lost this part of the temporal lobe has no problem with the landmark discrimination. He knows the answer in terms of where, that is, food next to the cylinder, does not know the answer to what, food under the triangle. If you lesion the monkey in the parietal lobe, you get the exact opposite pattern of deficits. And this monkey fails miserably at finding the food next to the cylinder. It seems like the easiest thing in the world, food next to cylinder, but this monkey is clueless. This monkey, the one who has lost a bit of his what system, has no problem whatsoever because he knows he doesn't have to discriminate shapes. Well, what about humans? Well, here, at least until recently, until the development of functional MRI, we had to rely on accidents or experiments of nature, cases in which patients came into a clinic having suffered a stroke, but they indicated the same what and where distinction. If there was damage to the parietal lobe, you got a condition called neglect. If there was damage to the temporal lobe, you got a condition called agnosia. It's basically the same word as agnostic, meaning don't know, but in this case it isn't that you don't know about the existence of God. If you're agnosic, you don't know what you're looking at. That is, I can place a comb in front of you and a spoon and a toothbrush and a cigarette lighter, and the person will not be blind. They will see them, they'll know their objects. They just have no idea what they are. Everything they look at is like handwriting in a language that they don't understand. They can see every squiggle perfectly, but can't connect them with their memory of what those things are. Now there's a special, actually let me first describe neglect, and then I'm going to give you a particular case of agnosia. Now, if you have a patient with the syndrome called neglect, caused by damage to the parietal lobe, you ask them to copy a drawing, in this case a clock, in this case a house, in this case a flower. You see something curious. It's as if the left half of visual space no longer exists for them. Now, it's not that they're blind in the left half of space. If they're sitting in a dark room and a light goes off in the left half of space and you say, "Did you see anything?" they say yes. However, left to their own devices, as they scan the visual field, they kind of forget that the left half is there. That is, they neglect it. So for example, they will eat only on the right side of their plate. They will shave or make up their face only on the right side. If you ask them to describe what's in the room, they'll only describe the things in the right half. If you then turn them around, so what used to be left is right, what used to be right is left, they'll now describe the things they forgot and forget the things that they described. And in the clinic, they can be tested by giving them, say, a clock, and you'll see that there is no detail on the left side of the clock. Or the house here has got two windows on the right side, nothing on the left. Flower doesn't have any petals on the left. If you simply ask them draw a clock from memory, you'll get a circle. They can just draw the whole circle in one motion. Then when they have to put the numbers in, then you go 12, 1, 2, 3, 4, 5. Now they know intellectually, abstractly, that a clock has got to have 12 numbers on it for the 12 hours. But because the left half of visual space is something they no longer attend to, they squeeze 6, 7, 8, 9, 10 in at the bottom right half, knowing that they have to be there somewhere, but not having an accessible representation of the left half of space. So that's a syndrome of neglect. Now the syndrome of agnosia sometimes occurs, depending on where the damage is, in a form called prosopagnosia, which means an inability to know faces, a particular subtype of aphasia. Here I'm going to show you a video of a man named LH who was in a car accident when he was a student. The car rolled over. His brain slammed into the bottom of his skull so that the bottom of the temporal lobes was damaged, which we now know to be the seat of the what system. And here he is being tested.
Do you have some picture in your mind of what those people might look like?
Not a particularly good one, no.
Recent research suggests just how important faces are to us as a species. We've evolved special areas in the brain that are set aside just for facial recognition.
There's evidence that there are distinct centers in the brain for recognizing faces and for recognizing facial expressions. And these seem to be separate from the visual centers in the brain that recognize objects in general. And evidence for this, for example, is that there are patients called cross-up agnosis, and that comes from where cross-up meaning face, agnosis meaning not recognizing or not knowing. Patients such as this who have difficulty recognizing faces that should be quite familiar to them, such as faces of their wife or their children or even themselves in the mirror. And these people, though, can recognize objects in the world. They can tell, apples from oranges or bananas. They can use utensils on their desk. They have the ability to recognize objects in the world, and yet they seem to lose the ability to recognize familiar faces. Take a look, for example, at this woman. Do you recognize her?
Lincoln Holmes lost his ability to recognize faces after a bad car accident 19 years ago. Now, although he knows this is a face and can read its expression, he doesn't know who it is. He can't recognize even the people he knows best.
One more here. What about right here? This man here?
No.
Any ideas from maybe from the context who that might be?
Not unless that's Martin Luther King, it's the March on Washington.
Work like this is exploring the complex connection between the brain and the face.
Can you just take a look in here and tell me what you see?
Well, since a mirror is the ultimate contextual cue, I know that's myself. However, it's important to note that as when I look at any other face, I am drawn to individual pieces rather than somehow seeing the whole thing. I am noticing my glasses, I'm noticing my hairline, little bits and pieces of the face like that.
Okay, but this man, by the way, Lincoln Holmes, completed his bachelor's degree at Harvard after recovering from the accident, went on to earn 2 master's degrees, and as you can see from his own explanation of his condition, the mirror is the ultimate contextual cue. This is a highly intelligent and intellectually sophisticated man, despite this rather striking deficit of misidentifying John F. Kennedy as Martin Luther King, for example. So this shows that Lashley was wrong in the extreme mass action interpretation that any amount of brain that you lose makes you duller across the board. It shows that there is some degree of localization in the human brain that allows highly preserved intelligence despite the loss of this particular function. But finally, let me say a few words about the frontal lobes, which are the part that humans tend to brag about the most, the part of the brain that tends to be associated with many of the higher intellectual functions of planning, decision-making, short-term memory, self-regulation, and so on. I'm now going to give you a few of the results that underlie that kind of braggadocio. For one thing, you'll notice that the frontal lobe in humans is quite mammoth. It occupies really the front half of the brain. And if you compare the relative size of the frontal lobe to the rest of the brain, and you scale for how big a brain and how big various parts you would expect given the body size of a primate, then you see that we really do have outsized frontal lobes, we as a species. We have bigger frontal lobes than you would expect for a primate of our size. This is a wiring diagram of the brain showing with the frontal lobe areas in black. Let me just zoom in on that. And just looking at the connectivity of the brain would immediately tell you that the frontal lobe is rather special, because all parts of the brain funnel information into the frontal lobe. So here you've got the auditory cortex, the hippocampus, which is responsible for consolidating memories, the amygdala, which underlies fear and other emotions, the insula, which is a part of the cortex that's involved in gut feelings, feelings of a full gut or a racing heart or a flushed skin, olfactory for smell, visual brain, the parts that we've been talking about, motor, somatosensory, the skin, all of them converge on the frontal lobe, which gives it a special role in conscious awareness and decision making. Namely, if you make it's the part of the brain where you can think about what you're feeling, what you're hearing, what you're seeing, and ultimately what you're doing. And so the inputs to the various other systems in the brain make it well placed for that role of deciding what to do. Also, let's go back here, it's not a coincidence that the frontal lobe is adjacent to the primary motor area. Why do we have the ability to think and plan? Evolution didn't design us just to be abstract cogitators, to prove theorems or to think lofty thoughts, but we come to intelligent conclusions about the world so that we can act on it. And not coincidentally, the parts of the brain that process information and that make decisions that are ultimately executed by actually doing something are the parts that are adjacent to the motor strip. And also, you can't really see this in the diagram because it comes up from the inside, but the parts of the brain that, the limbic system, the parts of the brain that underlie emotion, motives, feelings, urges, desires, cravings, also connect via the inside to the frontal lobe. So the frontal lobes allow you to plan and do in service of what you need and want. How do we know this? Well, we know this because the various parts of the frontal lobe have been associated both in monkeys and in humans with particular aspects of these high-level functions. And I'll briefly talk about three parts of the frontal lobe. Again, it's a massive structure. There isn't just one thing the frontal lobe does. The frontal pole, that's the part right in the front. The dorsolateral, dorsal again meaning upper, lateral meaning side, and orbital. You'll notice that there's a dent here in the brain. The reason that there's that dent is that's where your eyeballs fit. The bony sockets in the skull in which your eyeballs roll around are called the orbits. The part of the frontal cortex that sits above the orbits, literally on top of the eyeballs, is called orbital cortex. Okay, so dorsolateral frontal cortex is involved in your conscious thinking and problem solving, your working memory. How do we know this? Originally from an experiment done with our cousins, monkeys. If you give the monkey a test that depends on memory, that is, you show two wells of food in front of the monkey, you put a grape in one of them, you cover them both up, then a screen falls down so the monkey can no longer see either well. He's got to keep it in memory. Then his screen is removed. He's allowed to reach and uncover one of the two wells. Not a terribly hard task if you have an intact lateral frontal cortex. If the lateral frontal cortex is extirpated surgically, the monkey can no longer do this simple task. Now, it's not because he can't see or he can't reach, or even that he can't tell patterns apart, because if the food is always under the plus, so that he doesn't have to hold anything in memory, but he just has to keep the association, plus food, plus food, plus food, you don't need a frontal cortex to do that, and the monkey can solve this problem even after surgery that removes his lateral frontal cortex. Well, then there's the frontal pole. That is the anterior most tippy tip of the frontal lobes, which is involved in what neuroscientists call executive control of behavior. It almost sits at the top of the chain of command. It prevents you from acting on your immediate urges. It allows you to plan what you're going to do when, kind of the difference between being really an animal and a human. If someone has damage to the frontal pole as a result of an accident, say not wearing a seatbelt, being in a car accident, hitting their head on the windshield of a car, They will often have trouble controlling their behavior. They will be stimulus driven in that if you put a brush in front of them, without thinking, they'll grab the brush and start brushing their hair. If you put food in front of them, they'll immediately eat it. If they go into the shower, they might never come out until you pull them out because no part of them says, okay, you've had enough of a shower, it's now time to do something else. Finally, the orbital cortex, part above the eyeballs, and the medial cortex, I'm going to soon show you what that is, are involved in regulating motions, coordinating what you feel with what you do. Now, here we've got, as I mentioned, the orbital cortex. That's the part above the eyeballs. But remember, the brain, as a convoluted three-dimensional structure, can only be understood if you visualize it in 3D. This part of the brain is continuous with a part that goes kind of inside, in between the two hemispheres. In particular, the ventromedial frontal cortex. That is, we've removed one hemisphere here, you see the inner surface of the other one. This part of the frontal lobe continues into the orbital cortex, and they both kind of act together. Well, what do? The answer came from an unlikely source, probably the most famous patient in the history of neuroscience, a man named Phineas Gage. Now, this is an actual photo of Phineas Gage, recently discovered in a Vermont attic. You may notice two things about him. One of them is that his eye is closed. The other is that he's holding a long spike. Now, here is the story that connects them. In the 1840s, Phineas Gage was a railroad engineer supervising the laying of track in the rocky terrain of Vermont. The procedure at the time for laying track was to drill holes in the granite, put explosive powder at the bottom of the hole, then a fuse would go from the explosive powder out to the surface. There would be a layer of sand and a layer of sawdust that would hold the explosive powder in place. And then the fuse would be lit and a line of holes would all explode at the same time. Now, as Gage was supervising the work done by the workers, he had a steel tamping iron where he would compress the layers of explosive powder, sand, and sawdust, and he would compress them with the end of this tamping iron. Unfortunately, one of the worker had forgotten to put the sawdust and sand in one of the holes, leaving nothing but explosive powder. As Gage compressed the powder, a spark formed from the contact between steel and granite. The spark ignited the explosive powder. The tamping iron was well into the cylindrical shaft, and so it was fired out like an artillery shell, punctured his cheekbone, went out through the front of his brain and out the top of his skull, and landed 20 feet away. Here is this is Phineas Gage's death mask, showing the chunk of skull that had been blown off by the spike, as well as the damage to the eye socket where the spike had gone through. With this skull, modern neuroanatomists have reconstructed where the path of the spike, and by filling in the brain digitally with filling in the skull with the brain, what parts of the brain were likely to be damaged, leading us to conclude that it was the orbital and ventromedial frontal cortex that was damaged by the spike. Well, what were the symptoms? Gage never lost consciousness. He was awake the whole time. He was bundled into a wagon and pulled by horse to a hospital in Boston, 100 miles away. He also miraculously did not succumb to infection or swelling, but actually lived several decades after the accident. He was not blind, although he did lose one eye, as you saw from that photograph. He was not deaf. He did not lose his memory. But nonetheless, there was a significant change. And as the people who knew him before and after the accident noted, Gage was no longer Gage. One of the most succinct and informative statements in the history of neuroscience. That what he lost was the ability to regulate his emotions. And I will describe his condition and end this lecture using the flowery language of 19th century medical reports. And I'll end this way just to emphasize that the fascination of the study of neuroscience is not just that the mechanical functions of consciousness, the vision, the hearing, the ability to grasp, even the ability to memorize facts, is subserved by identifiable portions of the brain. But what makes us us? What gives us our personality? What allows us to make decisions? What allows us to make plans? What allows us to make sense of our personal narrative and translate them into future action? That too depends on circuitry in the brain. In this case, circuitry that heavily involves the orbital and ventromedial frontal cortex. So according to the attending physician, the equilibrium or balance, so to speak, between his intellectual faculties and animal propensities seems to have been destroyed. He is fitful, irreverent, indulging at times in the grossest profanity, which was not previously his custom, manifesting but little deference for his fellows, impatient of restraint or advice when it conflicts with his desires, at times pertinaciously obstinate, yet capricious and vacillating, devising many plans of future operations which are no sooner arranged than they are abandoned in turn for others appearing more feasible. A radical change in personality from a responsible young man, and the major lasting symptom from this rather extreme damage. So just very quickly go over some of the key ideas that I mentioned. The concept of neurons in the central nervous system, brainstem and cerebrum, distinction between gray and white matter, essential to understanding the brain, the theoretical distinction between mass action and localization, 4 lobes of the brain, sensory and motor strips, the role of Broca's area in language, the distinction between primary visual cortex and the where and what systems in vision, and finally the frontal lobes subdivided into areas on the lateral surface for working memory, the frontal pole, executive functioning, and medial and orbital cortex, integrating emotions and goals with thought and behavior. Well, that is maybe the world's quickest overview of the entire human brain, and I hope that you enjoyed and profited from it. Thanks for your attention.